So, if you've ever really thought about the fundamental machinery of life, you pretty quickly run into proteins.
Oh, absolutely. They're the essential workers.
Right. They do everything from catalyzing reactions to providing structure, regulating DNA, even fighting off pathogens.
But there's this massive catch, a catch that has, I mean, it's stalled biology for decades.
A protein's function is dictated entirely by its final three-dimensional shape, its structure.
Exactly. And we live in this age of genomics, right? So knowing the sequence of amino acids, that one-dimensional blueprint, that part's easy. We know billions of them.
But figuring out the final folded 3D structure.
That's the bottleneck. I mean, experimentally, using methods like X-ray crystallography or cryo-EM, determining just one unique structure it's a monumental effort. It can take months, sometimes years.
The numbers really do tell the whole story of this gap, don't they?
They do.
We have sequence data on billions of proteins, but the central database, the protein data bank, it only has about 100,000 unique solved structures.
Such a disparity, knowing the instruction manual versus knowing the final product.
So, okay, let's unpack this. The ultimate challenge, predicting that 3D structure from just the 1D sequence, it's known as the protein folding problem. And it's been an open research challenge for, what, more than 50 years?
Right. And that's exactly why we are diving deep into the source material from DeepMind today, specifically their paper, Highly Accurate Protein Structure Prediction with AlphaFold.
Which makes a huge claim.
A huge claim. It essentially says they've achieved atomic accuracy, predictions that are competitive with the structures that come from all that laborious lab work.
That is a tectonic shift, if it's true.
It absolutely is.
Yeah.
So our mission here is to really extract the core insights on how this neural network, AlphaFold, pulled this off, how it achieved this breakthrough performance, and what that means for the future of biology for you, the learner.
Okay, so to really grasp the significance of solving a 50-year-old problem, we have to look back. Look at the two traditional paths researchers took and why both of them ultimately hit a wall.
The first path was, you could call it the elegant but impossible ideal, the physical interaction program.
This involved trying to integrate all our knowledge of, like, molecular forces, thermodynamics, kinetics.
All of it into these massively complex simulations.
Intractable is the key word there, right? You're trying to model every single interaction between every atom at every moment.
Exactly. The computational difficulty was it was insurmountable, even for a pretty modest-sized protein.
And you're also trying to model the physics perfectly in the first place with all the chaotic effects of water and temperature.
The complexity just exploded.
So the field pivoted. It pivoted to the second path, the evolutionary program, or bioinformatics. This really capitalized on the explosion of genomic data.
Precisely. So instead of simulating physics, you look at evolutionary history. If a specific pair of amino acids have to interact in the final structure, well, evolution will correlate their mutation patterns across thousands of species.
And you can derive structural constraints from that, from looking at those correlations or finding a similar protein, a homologue that's already been solved.
Exactly.
This was a clever approach, but I remember it always felt like there was a glass ceiling.
There was. And here was the crucial limitation. These deep learning approaches, using evolutionary data, they were great at producing a rough general structure, a ballpark figure.
But they fell far short of atomic accuracy in most cases.
Especially if the target protein had no close relative already known in the PDB. They were, educated guesses. They weren't replacements for ground truth.
Right. So a useful starting point for the lab, but it wasn't a replacement for the microscope.
Not at all.
Okay, here is where it gets really interesting. Because AlphaFold essentially combined these two paths, but in a completely novel way.
What's so fascinating here is that AlphaFold didn't try to explicitly program the physics back in. No. Instead, it took the power of the bioinformatics approach, using those massive evolutionary data sets, and it incorporated physical and geometric inductive biases directly into the design of its neural network.
Yeah.
It learned the constraints of physics from the data itself, a much more scalable approach.
And the proof that this hybrid approach worked came in the ultimate gold standard for this field.
CASP.
The 14th Critical Assessment of Protein Structure Prediction, CASP-14, in 2020. This is like the Super Bowl of structural prediction.
It really is. And it's a blind test. The targets are protein structures that have just been solved experimentally but haven't been publicly disclosed yet.
So no cheating. They were truly predicting novel structures in real time. Give us the results, because the numbers here are just stunning.
So the main metric we use is root mean square deviation, or RMSD. It just measures the average distance between the predicted atoms and the true experimental ones. Lower is better.
And what did AlphaFold achieve?
AlphaFold's median backbone accuracy was 0.96 angstroms RMSD.
Okay. And the next best method?
The next best performing method in the whole competition was over 2 angstroms worse. It clocked in at 2.8 angstroms.
That is a massive, massive difference in atomic terms. So for you listening, someone who doesn't spend their life measuring atoms, can we put 0.96 angstroms into perspective?
Absolutely. Let's think about the basic building block of organic life, the carbon atom.
Okay.
The width of a single carbon atom is approximately 1.4 angstroms.
Right.
AlphaFold's accuracy of 0.96 angstroms is smaller than the width of a single carbon atom.
Wow.
It achieved near atomic resolution. At that level, you are essentially replacing months of difficult lab work with a computational result that is, for all practical purposes, experimentally accurate.
And the sources confirm this wasn't just some anomaly at CASP. This high accuracy holds up against a huge sample of PDB structures released after the training data cutoff.
And beyond that, the model gives you 2 things that fundamentally change how scientists can use these predictions: reliability and scalability.
Okay, reliability first.
For reliability, the model gives you these precise per-residue confidence estimates. They call it the PLDDT score.
And that is so critical. It tells the user not just the structure, but exactly which parts of it the model is confident about, like a core domain versus a floppy loop region.
So if you're a scientist trying to design a drug, you know exactly where the prediction might be a bit shaky and needs more experimental focus.
And scalability.
The method is fast, and it can handle really long chains. They showed an accurate prediction for a massive 2,180 residue single chain that had no known structural homologs.
So we've moved from years of lab work for one structure to
Yeah.
minutes of computation. Let's get into the engine room. How do they actually design a network that enforces physics without needing to simulate it?
The architecture is it's ingenious. It's a novel machine learning approach that consciously incorporates these physical and geometric constraints. It works in 2 main stages.
2 stages.
First, the trunk, which they call the Evoformer. It processes all the input data, the multiple sequence alignments, the MSAs, and all the pairwise features. Second, the structure module, which takes that refined information and generates the explicit 3D coordinates.
So the Evoformer is the brain figuring out the deep evolutionary relationships, and the structure module is the architect that builds the physically viable 3D model. Let's start with the Evoformer, because this seems to be where they merge the 2 historical paths.
That's right. The core idea of the Evoformer is to view structure prediction as a graph inference problem in 3D space. Connections are defined by residues being close to each other.
But the data coming in, the correlations from the MSA, that can be inconsistent, can it?
Very.
Like if the evolutionary data says residue A should be close to B and B should be close to C, but then A should be far from C, that's a logical contradiction if you want to build a single 3D shape.
Exactly. And that is where their big innovation comes in. The triangle multiplicative update and triangle self-attention.
Okay, that's a mouthful.
It is, but it's really the computational implementation of geometric consistency. The Evoformer is constantly updating the relationship between 2 residues, say A and B, based on what it learns about a third residue, C. It's mathematically forcing the data to obey the triangle inequality.
It seems almost, I don't know, counterintuitive to rely on something as simple as the triangle inequality in a cutting-edge neural net. Was that the big surprise here? That these simple geometric constraints were the key?
It wasn't simple to implement, but it was absolutely essential. It forces the Evoformer to produce pairwise data that can actually be represented in a stable 3D shape. It's the bridge between flat evolutionary data and solid physical reality. Deep learning enforcing structural integrity.
So once the Evoformer refines all those relationships, how does the structure module actually build the 3D coordinates?
So the structure module operates on a concrete 3D backbone. But, and this is really interesting, they initially represent this structure as independent rotations and translations for each residue. They call it the residue gas.
A residue gas. Wait, why would you break the constraint of the peptide chain? I mean, that's the most fundamental part of the protein. Doesn't that make the job harder?
It seems like it would, but it's actually a computational trick. It simplifies a notoriously difficult problem, loop closure. By momentarily ignoring the exact peptide bond geometry, the network can refine all parts of the chain at the same time without having to solve these really complex geometric constraints every single step.
That's a genius hack. So the final structure, only satisfies the real geometry after a relaxation step at the end.
Exactly.
So what's the key innovation in transforming that gas into the final, highly accurate structure?
That would be the invariant point attention, or IPA. Now, attention is common in machine learning, but IPA is geometry aware. It uses the 3D coordinates and a local frame for each residue, and the whole computation ensures the system is invariant to global rotations and translations.
Why is that invariance so critical? If the protein is folding, why does it matter that it's invariant to rotation? Isn't it fixed in 3D space?
It matters intensely because the folding process is iterative. The system needs to know that two residues are 5 angstroms apart, whether the whole protein is upside down or right side up. That local truth, the spatial relationship between neighbors, that's the only thing that matters.
So IPA ensures the network only focuses on the critical internal geometric connections, makes the process faster and more stable.
And the whole system uses this recycling process. It feeds the outputs recursively back into the same modules, refining the structure again and again, which hugely boosted accuracy.
And the final piece of the puzzle is the loss function they used for training, the frame-aligned point error, or FAPE.
Which sounds like a nightmare to calculate.
It does.
And it kind of is. It compares predicted atom positions to the true positions under many different alignments. Think of it like this. If you were grading a child's drawing, you don't just check if the nose is in the right place globally, you check if the nose is correct relative to the eyes, the ears, everything else locally.
So APE creates this strong bias for local correctness and makes sure the chirality, the handedness of the final structure, is correct.
Precisely.
So beyond the revolutionary architecture, they use some clever training tricks to squeeze out that last bit of accuracy. One of them mentioned was self-distillation. How did they use all that unlabeled sequence data out there?
Self-distillation is how they basically expanded their training dataset beyond the 100,000 solved PDB structures. They took a trained AlphaFold network and used it to predict the structures of about 350,000 diverse, unlabeled protein sequences.
So they created their own synthetic protein data bank, full of high-confidence computational structures.
That's a great way to put it. They filtered those predictions down to a high-confidence set, and then they trained the final model using a mixture of real PDB data and these computational predictions. It allowed them to leverage this vast amount of information that would have just been useless for training otherwise.
They also use a technique that might be familiar to people following large language models, the BERT objective.
Right, the masked MSA loss. Just like masking words in a sentence and asking a network to guess the missing word from context, they randomly masked or mutated residues in the multiple sequence alignment and forced the network to predict them.
And that helps it learn the deep phylogenetic and covariation relationships needed to reconstruct the structure. It makes it incredibly robust.
Exactly.
Okay, it sounds like a silver bullet, but let's talk about where it still struggles.
Yeah.
Where does AlphaFold's predictive power start to break down?
The source material identifies two main areas of vulnerability. First, it is heavily dependent on evolutionary context.
Meaning the MSA.
Right. If the median alignment depth, the number of effective sequences in that MSA, is less than about 30, the accuracy drops off substantially.
So if a protein is evolutionarily unique, or if we just haven't sequenced enough related organisms, the Evoformer can't gather enough data to find those patterns.
Precisely. If you don't have that evolutionary context, the network just struggles to derive the fundamental geometric constraints. The games start to plateau around 100 sequences, but you need that minimum context to even get started.
And what's the second limitation?
AlphaFold is significantly weaker for proteins that have many heterotypic contacts.
Heterotypic, so interactions with other chains in a complex.
Yes. Like when a protein only folds correctly when it's interacting with a different partner protein or maybe a piece of DNA or a ligand.
And that's a problem.
It's a big problem. AlphaFold, as it was trained, is primarily designed to produce the structure of the monomer, the single chain that is most likely to appear in the PDB. If that chain is inherently unstable outside of its complex, the prediction might not reflect its true functional biological state.
So if we connect this to the bigger picture, even with those limitations, the ability to rapidly predict structures at this level of accuracy, it means structural biology can finally accelerate to keep pace with the genomics revolution.
The speed and accuracy mean prediction is now possible at the proteome scale. Just imagine predicting the structure for every single protein in the human body. And we know from the source material that AlphaFold is already being used by experimentalists for things like molecular replacement or interpreting really challenging cryo-EM maps. It just removes one of the most frustrating bottlenecks in molecular biology history.
This has been a true deep dive into a monumental achievement, one that has, I think, restructured an entire scientific field overnight. So to summarize the core insight, AlphaFold succeeded by merging the two previously limited approaches, bioinformatics from evolutionary history and physical modeling from geometric constraints, and packaging them into this recursive, deep learning architecture.
An architecture that achieves near experimental accuracy in minutes. It accelerates structural discovery from years to minutes. And it's hard to overstate the impact of that acceleration on future medical and biological breakthroughs.
So what does this all mean? We've moved from a scientific problem where solving one structure took years to a computational prediction taking minutes, opening up possibilities we just couldn't access before. Our source material noted AlphaFold's limitation when dealing with interactions between different protein chains, those heterocomplexes. So here's a thought. If the powerful methodology AlphaFold uses can be readily adapted to accurately predict the structures of full multi-protein assemblies and protein-ligand interactions, what fundamental biological mechanisms, currently a structural black box, might we be able to visualize next, allowing us to finally understand diseases like cancer or Alzheimer's at an atomic level?